just note that we're now recording. If you don't want your face to accidentally appear on video, feel free to turn off your video, but we always like to keep at least a few faces on for the speaker so they don't feel alone. So if you don't mind, just keep your face showing and that's great. So Paul Jaschke is our speaker. He is an assistant professor of synthetic biology at Macquarie University in Sydney, Australia. And he's also the CSO of Hyperdrive Science, a startup that uses phage display to help pharmaceutical companies. And so I think a cool factoid about Paul is that I found out he went to the same university I did in Edmonton, Alberta, Canada. Here I thought I was inviting an Australian phage researcher and he's secretly Canadian like me. So that's the fun fact, but he's going to tell us about engineering phages and we're super excited to have him here. So yeah, I'm going to turn it over to you, Paul.
Thanks very much, Jessica. Maybe we even have the same accent. I'm not sure. I can't even tell anymore.
Well, we can't tell. No.
Yeah, definitely not. If you, we're expecting an Australian accent, you'll be sorely disappointed, but hopefully still stay for the science. Yeah, so it says lecturer. I always say assistant professor because that's more understandable. It's the same. A lecturer is the equivalent in Australia. Yeah, I'm in the molecular sciences department in Macquarie University. Thank you very much for the invite. Jessica and I have, I think, corresponded a few times over the last year, maybe, when I found out about phage directory and PHAVES. And so, yeah, it's really great to be on here for the opportunity. So thanks very much. Okay, so I'm going to tell you a few stories about the research that I've done on phage. I think hopefully it'll be diverse enough. It'll interest, you know, at least a little bit for everyone. I'm going to start just by talking a little bit about the lab. So here we are at the top of the world, not the bottom. So I've decided to flip the map here in beautiful Sydney, Australia. And so this is most of the lab here. We have a master's student who's joined Pamela, who's working on phage as well. And you can see by Bradley here, a lot of his research is featured. You can see my cursor, right? Yeah, we love phage and he's got this great phage t-shirt. So he just recently got his PhD a few months ago and is looking for, actively looking for postdocs. So if you have something available, please let him know. It's been a bit challenging with the pandemic corresponding with the postdoc job search, but he's been doing well. And so, yeah, we're at Macquarie. We have our own little lake here with lots of cool Australian birds. So it's a beautiful spot. If you ever get the chance in the next few years when things get back to normal, I'd love to have people come visit. So let me know. Okay, so onto the star of the show, my favorite bacteriophage, Phage 174. So I started working on this in my postdoc in Drew Endy's lab at Stanford. Previous to that, I'd never worked on bacteriophage, and I was just talking with Stephanie about you know, being a lone phage researcher. I wasn't the lone phage researcher, but I was the only one working on Phage in the lab, and the only one who had ever worked on Phage in the lab, to my knowledge. And so Phage is sort of, you can consider a model for the Microviridae family. And they're seem to be dominant and persistent members of the human gut virome, shown in this from a recent paper. The relative abundance in the persistent group is quite high, as well as the transient, although it seems to have a wide variation. And so Phage in particular infects a type of E. coli called E. coli C, which has this rough outer core lipopolysaccharide. It also infects E. coli W strains. I think I'm the only one that knows that, which have similar sort of LPS types of this R1 to R3, but it cannot infect K12 type. So that is always the pitch I use when we're working on this in labs where people are also doing a lot of cloning, is that don't worry, our phage isn't going to kill your cloning strains. It can also infect Salmonella. And Phage seems, based on some of the work from this Michel et al. Paper in France, seems to exclusively infect human gut-associated E. coli strains. So although it maybe isn't that prevalent in the human body in terms of numbers compared to some of the other phage, the PHAVES anyways, it seems to be quite important. Diving down a little bit more about its infection process. So you have a particle here. The kind of green color here is the gene F, protein F, which is the major capsid protein. And in tan is the protein G, which is the spike protein. And you can't see two of the proteins, which are J, which helps hold the single-stranded genome inside the particle, and H, which is the pilot protein, which has only recently been shown to be a just-in-time tube to transmit the genome inside the cell. And so what happens is during the infection is the spike makes contact with the lipopolysaccharide, and we get the spike proteins dissociating on one of the vertices. And then you get emptying of the capsid with the H protein creating this DNA conduit shown here. And so this is a cryo-EM picture with the electron density in pink here of a full capsid, and in blue of the empty capsids. You can see where the single-stranded genome lies and then gets injected through this tube in this hole. And I realize this is not needed for this audience maybe, but I have this for other groups. This is a plaque of PHAVES on a plate here. And this is a little video of PHAVES grown on minimal media and then watched for 3 hours. So the infection happens right at the beginning, and you can see that there's still replication of the bacteria for a while, but then all of a sudden the lysis occurs. And PHAVES creates a protein called protein E, which is an antibiotic-like lysis protein. And so it disrupts peptidoglycan synthesis. And I'll talk a little bit more about that in a minute. But looking at the genome, which to me is probably one of the more interesting aspects of PHAVES, it's 5,386 nucleotides long. It's circular single-stranded DNA, and there's 11 known genes, and 9 of these are essential. So the circular view is here. But one of the aspects that's really interesting about PHAVES is prior to sequencing, if you totaled up all the genetic material necessary to encode the proteins of PHAVES, the researchers realized that you'd actually need a longer genome than they knew that PHAVES had. And so there's this sort of mystery. How can this virus encode more protein sequence than it has genetic material to do that? And once they sequenced it, they realized what the answer to that was, which was there's an extensive gene overlap. And so, for instance, gene A here, we have the gene B completely embedded within that gene A in a different reading frame. And likewise with the E, the lysis protein is completely embedded within gene D here. And we have a lot of also some stop and start codon overlaps in some of these other regions. And so people, when they first sequenced the genome, said, wow, this is amazing. You know, what could be the reason for this, for all these gene overlaps? And I think my favorite explanation for what that could be is that group in Japan sort of explored the idea of, could it be a message from extraterrestrials? So their thinking was, if you're going to send a message across the stars, it's good to encode that message in sort of a universal material. Maybe DNA is universal storage material. And as well, if you want that message to not be corrupted over time, you have to put some sort of error correction in that. And maybe these gene overlaps were that. So their thinking was, maybe in the areas where the genes were overlapped, any mutation would affect more than one gene, and therefore have a higher chance of disrupting the phage and stopping its life cycle. And so by encoding a message in these gene overlaps, maybe they've sent us a chart, a star chart or something like that. And so they started looking at these regions of overlap and trying to figure out what code might be encoded in there. And here's a figure from the paper, but in sort of a refreshing honesty that you don't see in papers very much anymore, they concluded at the end of the paper that we weren't able to find anything. All right, but probably a more likely reason is this shown here. So the infectivity on the y-axis and the x-axis is the size of the genome. And so here's the actual size of the PHAVES genome. So if you increase the genome size, even up by a few base pairs, the infectivity just drops off this cliff. And so if new genetic innovation, new genetic function is going to be encoded in this genome, it has to use the base pairs that are already there. And that's probably where these overlaps came from. So before I get talking a bit about the engineering process, I'm going to maybe talk for a few minutes about some recent results we had, just trying to characterize the actual infection of these microviridae, because all the research that had been done on this virus had basically been done from the 70s, 60s and 70s. And although that was done by very able researchers, the resolution of some of the things they were doing was just not as high as modern methods afford. And so Bradley Wright and Dominic Logel, 2 PhD students in the lab, undertook doing some omics analysis of the Phage infection. And so looking at what proteins and RNAs are created during the infection. And one of the things we found was that these heat shock chaperones called the IBPB and A genes were highly upregulated during infection. On the left is the protein volcano plot, and on the second here is the RNA plot. And the IBPB and A genes are upregulated to the same level as some of the Phage proteins. So they're highly expressed during the infection. And so we tried to understand what the role of these proteins are in the infection. And we think the reason the transcripts are so much more highly upregulated than the proteins are that there's actually a thermometer function in the RNA, so that it's only highly translated at elevated temperatures, because it is related to heat shock. And that wasn't present in our studies. So we got a lot more RNA than protein for those. And so just taking a look more at this heat shock chaperone pathway, we have a protein in the middle here. And then we have a normal folding pathway here, creating a nicely folded phage protein. But if too much is being created, the cell senses that and starts grabbing onto it using these holdase proteins, which hold it in this nearly folded conformation, but not quite. And then that's released later with the ClipB protein. And you can see most of the proteins in this pathway are all quite upregulated during infection. So what we think is going on is the Phage particle is attaching to the cell. This is, I think, approximately true size, true representative size, injecting its genome, and then that's starting to pump out a whole bunch of protein, more than the cell can fold. And so the holdase proteins bind to that protein, bind to those almost folded proteins and hold them there. And then the ClipB protein which is highly upregulated as well, will release that and then allow the folding and particle maturation to occur. And if you think about it, this is a nice system because these proteins are not aggregated. They're held in an almost folded position and are only sort of being released in sort of a controlled manner by the ClipB protein, allowing a sort of steady stream of phage to be created in an efficient manner. And then the second interesting thing we found was related to that antibiotic-like protein, protein E from Phage. And so this is the lysis protein. And so how this works is it binds to this mRNA protein in the inner membrane and disrupts the first membrane step of peptidoglycan synthesis. So peptidoglycan is important in gram-negative bacteria because it's this layer in between the outer and the inner membranes. And so what we found was that in the infected cells, all the proteins in the steps prior to where protein E interacts with mRNA were not upregulated, but everything downstream of that was. And so it seems like the cell is sensing that it's not making enough peptidoglycan, and it starts upregulating all the proteins involved in peptidoglycan synthesis in the membrane. And in particular, also a lot of the membrane transporters involved in lipoprotein trafficking to the outer membrane were also highly upregulated. In particular, this protein LPP, which is an anchor that helps anchor the outer membrane to the peptidoglycan layer. Okay, so that all seems to make sense. And so that was sort of learned as we were trying to learn more about Phage's function. And so now I'm going to shift a little bit towards using part of the talk. So using phage genomes as models for synthetic genomics. So what exactly is synthetic genomics? Well, it's the extensive and intentional genetic modification of a replicating system for a specific purpose. So this is from a review by George Church and Peter Carr a few years ago. Some of the things you can do, sorry, I'm going to have to go shut the window here. Just hold on. Sorry about that. Somebody decided to do some gardening next door or something. Where was I? Yeah, anyways, so synthetic genomics. So some of the interesting things that people have been doing are, so for instance, reducing the size of the E. coli genome and seeing some interesting emergent properties there. Some really interesting things have been done in that space with virus attenuation across a whole genome, so changing the codon bias and things like that. And as well, the total synthesis of E. coli's recoded genome, and also this work a few years before that, trying to reduce the number of codons in E. coli below 64 so that you free up some codons in the genetic table for, say, expressing an unnatural amino acid so that you can, say, add new chemistry to the genetic code and things like that. But one of the, why would you use phage genomes to address this? Okay, so I think of them as these self-contained genetic programs that runs within a constant cellular operating system. And so that way, it's easier to change the phage and to see if it affects some point of its life cycle or breaks the life of the phage. I keep using life, but breaks the cycle of the phage replication. Whereas if you did this to the cell and it was no longer viable, it's sort of hard to rescue that because it's not living anymore. And it also sort of occupy this sort of sweet spot between a very simple protein producing gene cluster and a full cellular genome. So they have some complicated aspects to their life cycle. And depending on the phage, you can go from very simple life cycle to very much more complex. And they also can, say, regulate their genes in certain ways. And of course, learning about how to engineer phage genomes better is also quite important because they, I think, are a really interesting potential next generation antimicrobial therapy. And so by learning what we can do with their genomes, could potentially help in that area. So one of the major issues when you're making changes across an entire genome is what do you do with the overlap genes? And so PHAVES is unique, not uniquely positioned, but very beneficially positioned to address this. And so if you start making, say, changes to remove codons, what if some of those codons occupy this space and are then going to affect this overlapped gene? So I'm not sure if people know, but gene overlaps are very prevalent across all the cellular domains, as well as being extremely common in viruses. And my student, Bradley, just sent me a paper saying that somebody had just published something about an overlap gene in SARS-CoV-2. So it's everywhere. And so in E. coli, you have the menaquinone pathway, for instance, having some overlap genes. And as well, it's becoming more and more appreciated even in higher organisms like eukaryotes and humans. We have extensive gene overlap. So for instance, the NF1 neurofibromin gene has this very long intron where 3 other genes are embedded in there in the reverse direction. And so what are these gene overlaps doing? Well, in prokaryotes, it's believed that some of the things that they're doing are, for instance, when you have a stop codon, start codon overlap, the ribosome that's translating upstream is actually potentially unwinding some RNA structure that's allowing the downstream gene to be more efficiently translated. So this can be thought of as translational coupling, and as well this dissociation of one ribosome and a reinitiation of another one is also thought to be a reason for that. And then related to what the Japanese researchers thought was maybe a role for the overlaps in PHAVES is this fact that when you have overlap genes in different reading frames, any sort of single point mutation can potentially disrupt the amino acids of multiple genes. And this can be quite important in PHAX because PHAX has quite a high mutation rate, relatively. All right, so to address this idea and to look at how important and what can we do when we're rebuilding genomes to address overlap genes, one of my first projects when I was in the ND lab was to decompress or remove all the overlaps in the PHAX genome. So on the top is the wild type genome with the start-stop codon overlaps, and on the bottom is this genome that I designed and built called calling the decompressed PHAX. And so, for instance, we take gene B that's embedded in A, and we move it downstream of A, so it's no longer separated, so it's physically separated. And again, the stop and start codons are now physically separated. And I tried to put them in as close to position as I could in the wild type and in the same order. And so K, instead of being embedded in A, now goes downstream of A, and likewise with C and D and E. I'll tell you about F in a second here. But the same issue that we faced with decompressing PHAX, you can see when you're trying to refactor protein-producing clusters, say from the Klebsiella oxytoca with a nitrogen fixation cluster from Chris Voigt. They did the similar sorts of ideas. They had start-stop codon overlaps, and they had to then remove those genes and separate them physically. And likewise, in that most recently recoded E. coli project, there was also overlaps that they had to deal with as they were trying to remove these codons. So it's quite a relevant topic to a lot of different types of engineering projects. And so how did I do this? So just a different picture of this. So if I had unoverlapped everything in the PHAX genome, it would now be 6,310 base pairs long, which, as that figure showed before, would now take us about 5 logs down in terms of infectivity. It would really make it a really terrible virus, probably. And so instead of doing that, I took part of, I basically left a small fragment of gene F, which is the capsid protein in the genome, and moved that to be expressed in trans off of a plasmid. So this sort of gives a level of biocontainment as well, I guess you could say, because the virus is not able to replicate fully in cells that don't contain this gene F plasmid. And so after creating this phage inside yeast, I transformed it back into E. coli, and you can see that the plaque sizes here are quite a bit smaller than the wild type. And so that sort of sat for a few years until a PhD student, Bradley Wright, decided that he was going to start doing some more detailed measurements of the phenotype of this phage. And so one of the things that he found was that, yes, it has smaller plaques, but that if you infect cells at a higher MOI, it does seem to lyse cells at the same efficiency as wild type, which was interesting. And we were sort of wondering, well, okay, why does it make the small plaque size? Oh, I should mention it also had a smaller burst size. And so that made sense. So we wanted to say, is there something different about the capsid? You've changed the single-stranded genome, which we know is quite important for the packing of the capsid. Is that something going funny with the structure? And so we did some cryo-EM with collaboration with UNSW, and we found that the structure of the capsid seems to be within the measurement error, seems to be indistinguishable. But an interesting thing you can see from this TEM image is that whereas in the wild type from a cesium chloride gradient band, you get all these nice, perfectly formed capsids or virions, in the decompressed, you don't get as many, and you get some weird aggregations and things like that, which we're still trying to understand. But for the ones that are fully formed, they seem to be properly formed. So then we're trying to understand why is it a worse phage? What's going on during the infection? So Bradley did some very detailed proteomics analysis of the infection cycle. And I'll just give you the highlights of that. But essentially what he found was that one of the proteins, protein B, which is an internal scaffolding protein that helps the phage basically assemble at the very early stage in the procapsid, the expression of that protein was really decreased. And so you can imagine how that might mess up the infection, the assembly of virions. And just to give you context of what we did with gene B, we moved it from being embedded in gene A to then being downstream of gene A in its own separate coding sequence. And likewise, we also found a major difference in protein C, which is highly downregulated during the infection. And that protein is quite important for switching between stage 2 and stage 3 DNA replication. So stage 3 is where it's starting to put single-stranded genomes inside the capsids. And so gene C was less perturbed than gene B, but it still had a stop-start codon overlap here with gene A disrupted, and was now had on its own separate coding sequence from K. So to summarize, I didn't talk about it, but it had smaller burst, but also had poorer attachment to cells. We're still really trying to understand why that is. And it seemed to have quite highly perturbed protein expression, at least in a couple of the open reading frames. So take homes from that are 5' untranslated regions seem to be very important. Maybe that's a duh, but yeah, you can't naively remove gene overlaps and hope that fitness is not dramatically impacted. And also changing the transcript boundaries may have had dramatic effects. We're still going to investigate that further. And in future engineering, protein measurements may be necessary during this tuning process. Things we're still trying to understand is this static versus dynamic virion function. So maybe the virion's structure, when you just look at the static cryo-EM image, is the same, but maybe its dynamic function is impaired based on the different genome sequence, or based on, or possibly based on different protein stoichiometries in the capsid or things like that. And can we use semi-rational ribosome binding site tuning to restore the fitness, or is there something even more fundamental about those stop-start codon overlaps that we've disrupted that's just not able to be recovered? So work continues in that domain. So shifting gears now to another aspect of engineering genomes. This is the idea of cryptic genes and our sort of dream of finishing the genetics of Phage 174, which hopefully I'll be able to describe properly. So another aspect when refactoring genomes and gene clusters is that when you're trying to move these genes around to restore some sort of function, so in this case, it's the type 3 protein secretion system, they started moving genes around from its host organism to another organism, and found that, using all the genes that they knew were the ones that were involved didn't allow it to function. And so what were they missing? And what they realized after a lot of probably hard work is that there were the annotations weren't sufficient in the genomes, and there was actually 2 genes where they thought there was 1 gene. And so this is going to be an increasing problem when you're starting to refactor or redesign entire genomes, is you're only as good as your genome annotation. And so that got us thinking. You can think of the Phage genome a couple different ways. One is this very simple version that we see in the textbooks mostly, where the 11 genes that have been experimentally observed are here, and they have this size range, and everything's good. But if you start actually looking at the code and all the coding possibilities, you start seeing a lot more complicated picture. And so if you start looking at both strands in all 6 reading frames, and looking for any open reading frame that could be greater than 60 base pairs long, you start seeing many 326 in this case. But their range and size is actually quite similar to what's already been observed. And so you say, well, maybe there's more there, or how would we establish if there's not more there? And so we took a genome synthesis approach to that, coupled with bioinformatics. And so we looked at which ones of these are conserved across the microviridae. Let's focus on those. And we got this smaller subset, I think it was around 80 or 90. And then we said, what could we do to disrupt these? And then taking as our basically hypothesis going forward is none of these encode any function, but how can we prove that? And so we did that through basically disrupting them or erasing them. we're able to do that for most of them. Unfortunately, some of them are overlapped still with this highly overlapped region here. So this was done in the wild type background for various reasons that I can go into later if you want to hear the full story. And so ones that are in the region of highly overlapped, you couldn't make any changes that didn't disrupt the other known genes. And so one of the goals of this was to not change amino acids of any of the known genes, because then we would have a hard time telling what was going on. So with this design, which we called Clean X, because we cleaned it up, shows here all the changes that I made to the genome, including deleting or disrupting start codons, putting in stop codons to prevent the translation of these potential cryptic open reading frames, as well as weakening ribosome binding sites. So after that, I synthetically constructed the phage and then booted it up inside E. coli and found that, lo and behold, I'd made a worse phage again. Its plaque size was reduced, but not quite as much as the decompressed. And I tried to understand that by making a set of chimeras. So I split the genome up into 5 pieces of approximately equal size. And then I made a set of chimeras where it was either the wild type piece there or the cleaned up piece there. And I made a set of all of those combinations. And this is shown here for each of the genome sections. It's either green, which is the wild type, or red, which is the Clean X version of that. Sorry if this isn't visible by colorblind people. I'm not sure if this is colorblind friendly or not, but you can see here the Clean X genome is right here. And on the y-axis here is plaque diameter. So this is a rough measure of fitness of the phage. And so you can see wild type sits here, and within our measurement we can make slightly better ones, but Clean X plaque size sits here, and we could. One thing we notice is that all of the phage with part number 5, shown here as the Clean X version, had much smaller plaque sizes. But to try to understand, and so over here you can see that part number 5 corresponds to gene H, which is that pilot protein that we talked about before. It's not actually expressed at that high of a level in the phage, and does seem to be fairly robust to changes in some of the subsequent work we've done. But it looks like something's going on in this section. Now, the one thing we couldn't discriminate was, well, this section actually had the most nucleotide changes from wild type, and that was sort of spread throughout. And so how do we sort of discriminate what's going on there in a better way? And so the method that I hit upon with some help of a few undergraduate students over a summer is we allowed evolution to sort of show us the way. And so we took the host strain, E. coli C, and we took the Clean X phage and we infected it and allowed it to grow for a few generations. And then we took the lysate from that, added fresh E. coli and took some of that phage, and then did that over and over again for 48 generations. I believe we got the method for this from Holly Wickman's work on Phage evolution. So that was much appreciated. And after doing that several different times, we got the same single base pair mutation in gene H. And that corresponded to where the Clean X had a C in that position, the wild type had a T. So we changed a T to a C. And the reason we did that was to erase a start codon of this open reading frame number 36, which was one of those 300 that we had identified previously. And in both cases, we got reversion back to the wild type sequence. And we also got reversion back to wild type fitness and plaque size. And so we said, well, what's going on there? Is open reading frame number 36 a vital protein for Phage replication? Did we discover a new gene? Or did we do something to gene H? And so if you look at that region of gene H, it's actually the third codon of gene H, and we changed a glycine codon from one to another. So we didn't change the amino acid that was encoded, but we did change the code. Whereas open reading frame 36 here, it's a GTG start codon, and we change it to a GCG, which is not a recognized start codon in E. coli. And so we did some proteomics of the phage, and we also did some other in vitro tests. We were not able to detect open reading frame 36 protein using proteomics. And then we also, and then the other piece of information we got was using the synthetic gene H variants that we expressed in a cell-free system, and then looked at fluorescence, the amount of translation essentially from this. We saw that the amount of translation of gene H into protein was greatly reduced in the Kleenex background. And then when we made that one base pair change that was reverted in that phage evolution experiment, we got reversion back to about a doubling of protein production, not nearly back to wild type, but we did get about a doubling. And as well, if you look at the in silico folding of the RNA around the gene H start codon, in Kleenex, we made it a lot more stable structure, which is generally assumed to have less translation from it, from wild type. And that reversion sequence also made it a little bit closer to wild type, although it's still quite stable. And so at this point, we concluded that it was likely not from disrupting ORF36, but in fact from disrupting gene H translation. And so that's where the story ended, where we were trying to definitively demonstrate that by using DNA synthesis and design, you can basically establish that there's nothing more to know about a genome. I think we got most of the way there with this work. Okay, so to summarize what I talked about today, we've done some work doing some basic biology characterizing the microviridae infection. I find we tend to do this because we want to understand the system we're engineering, and there's always going to be information gaps. And so we want to do some basic science along the way to learn, how these systems work. And then also told a little story about decompressing the Phage genome, and so some of the perhaps a cautionary tale of some of the things that can go wrong during that process, and how you can use proteomics and to sort of nail down what are the exact problems. And then the last one of cleaning up the cryptic genes from a genome. And so I think I'm fairly convinced after a lot of the other proteomics that's been done by Bradley in the lab that there are actually no other proteins in Phage being expressed during an infection. And so maybe, I don't know if people know this, but the Phage genome is actually used in Illumina sequencing as a control. And so maybe it could also be used as a sort of protein translation control as a system with no other cryptic information encoded in there, but who knows? Okay, so just a very quick, what's coming up next soon-ish. I like the ish part because it could be a few months, could be 6 years, who knows? Alina Trofimova was a master's student in my lab and was quite talented in terms of her computer skills, was frustrated with current offerings of plaque quantification tools. And so she's building her own, or built her own now, and hopefully we'll publish that one of these days. It's hard to see here, but the system will identify plaques that are separated from others. It will sort of throw away plaques that are overlapped with others, like in this case. She's also told me that it can deal with the dreaded fried egg morphology of plaques. So Phage plaques tend to have these black centers in the middle, which really mess up some of the other phage plaque counting systems. And so hopefully it works. She's been using some published pictures, and you can see that some of the pictures don't start off super great quality. And so she's still able to get some good data out of there. So yeah, stay tuned. Also, we've got some really interesting stuff going on with phage biocontainment. I can't really talk about it now because it's still like very early stage and part of a person's master project, but we're trying to make phage that are a little bit safer to use outside of the lab, because I think a lot of benefits could be gained from engineering phage, but it's going to be a long road to making people comfortable to put those in their bodies, I think. And also a new project that's just getting going. I think I saw Shala, who's a PhD student who's starting this project, trying to tackle urinary tract infections with phage. So UPEC bacteria are these uropathogenic E. coli that cause a majority of the urinary tract infections and are really nasty, especially with the recurrent ones. And so we're trying to harness some of the power of these things called the Phage biofoundry to hopefully construct some massive libraries of these phage to tackle some of these problems. And as well, a really interesting project I have going with Drew Endy, where we're trying to use phage genomes again as these sort of in-between cellular and simple protein systems to trial what happens when you start really messing with the genetic code. Can you encode a phage using a highly reduced set of codons and still get a viable phage? And this is with the ND lab, and John Cowles is the PhD student who's working on that. Okay. Thanks to everyone, especially ND Lab, where some of this work, majority of the early work occurred during my postdoc, and some undergrads, Adrian, Sierra, and John Rodriguez, who helped publish that paper on the decompressed phage, and as well Erica Lieberman, who's now working in France with Eligo Biosciences. She was an author on that decompressed paper as well. And the definitive demonstration, so Cryptic gene paper was Diane was actually a high school student when she was working on that. And she's now a software engineer in the Bay Area. So that's great. This is Kay's interpretation of our work relationship during that summer of doing all these plaque assays. Apparently that's me. And Gabrielle Dotson is a very talented undergrad who's now doing her PhD in computational bioinformatics in Michigan. And then my lab, Dominic Logel, did a lot of the transcriptomics work and has recently published a cool paper that I didn't have time to talk about, but where we look at the transcription in PHAVES using next-gen sequencing and discover possibly some new promoters and terminators. And Bradley Wright did all the proteomics work. There's Alina for that phage tool, plaque tool. Bradley Wright did all the proteomics and for the ACS and bio paper, as well as this paper that we recently have on bioRxiv with all the omics of the host response. That was in collaboration with Professor Mark Malloy, who's at the University of Sydney, and APAF, which is the Australian Proteome Analysis Facility, where we did all that work. And as well, the cryo-EM work was in collaboration with Joan Fang Ruan at UNSW. I think that's all I got for now. So thank you very much for your attention. Hopefully I wasn't checking the time. Hopefully I stayed mostly the time. And yeah, happy to answer any questions you might have.
Awesome. Thank you so much, Paul. That was wonderful. And yeah, I think we all learned a lot, and there's lots of excitement in the chat, especially about this plaque quantification tool. I think it's like stealing the thunder. So we need to know more about this in the future. Yeah. I'm going to just call on a few people that have asked questions. And if you hear your name, you can unmute yourself and ask the question, and I'll give you a little bit of heads up. So we're going to start with Shanmuga, and then next will be Adriana. So Shanmuga, do you want to ask your question?
Sure. It's a very nice talk. I really enjoyed the talk. My question is, when you're looking at the plaque sizes, have you measured the burst size in any of these versions to compare it to wild type so we can get some quantitative information?
Yes, we did measure the burst size, or I should say Bradley measured the burst size of the decompressed phage. And so the normal PHAVES burst size is around 180, and in the decompressed it's around 18. So it's about 10-fold less. It's really a terrible virus. And in the CleanX, no, I never did. I was finishing that work right as I was finishing my postdoc, and didn't continue it once I got to here. So, so no, I never actually did the burst size of that one. I would imagine it's probably about 60%, you know, just based on plaque size, which somewhat correlates, I think. Maybe, yeah, 60% of the wild type, but I don't know.
Thank you. Thank you very much. Yeah.
Thanks for your question.
Awesome. Okay, Adriana, you want to go next?
Hi, thank you for your talk. So I have a question. Have you looked at possible interactions between the capsid protein and the phage genome?
Have I looked, the last little bit cut out there, but have I looked at binding interactions between the capsid and the phage genome? Or sorry, correct, between the capsid protein and the phage genome? I have not. I think the only person who has, I believe, is Bentley Fain in Arizona. And so the gene, it's gene J, which produces protein J, is kind of decorates the inside of the capsid and interacts with the genome. But no, I have not. I've been thinking about doing that, especially in the decompressed.
So the reason, sorry, I cut you off. The reason I ask is because here in the CPT, there was some work done in Phage Q beta, and it's a RNA phage, but in the cryo-EM of Phage Q beta, you clearly see interactions between the capsid protein, the inside of the capsid protein when it folds, and the phage genome. So if those interactions do not happen, then you won't have a regular capsid with a regular packing of the genome inside, which could explain all the phenotypes that you're observing.
Right.
So
yeah, I don't think our cryo-EM was high enough quality to see that. I was thinking actually of trying to look at it using some like Raman spectroscopy and things like that. I don't know if you've done anything like that, but just to look at the different conformations of the DNA.
So the thing is that, from my understanding, it's because it's a single-stranded RNA molecule. It'll have, so the constraints of like the biophysics and the constraints of that molecule inside of the capsid are going to be given by the space between it, like inside of the capsid, and which is going to be modulated by the interactions of that of the proteins that are going to wrap the genome itself.
Yeah.
That and then based on the amount of mutations that you've done, I think, so my take on this is that it doesn't seem to be very strict on terms of the sequence that these proteins are interacting, like the capsid proteins are interacting with, but maybe the secondary structure of your single-stranded DNA might be determining the way the protein interacts with the genome. Have you looked at secondary structures in the places that you've done mutations at, and maybe the changes in your EMs?
We've definitely done the sort of in silico secondary structure prediction, and yeah, I mean it's like completely different than the wild type in many regions, but we sort of were just like a little bit like, you know, it's in silico. DNA, I think, has a lot more constraints on how it can fold compared to RNA, for instance.
Correct. Yeah.
And like you were saying, I believe in PHAVES, right? The protein J also has sort of a, believed anyways to be non-specific binding, just more of a charge complementarity as opposed to a sequence specific sort of binding characteristic. But yeah, I would not be surprised at all if we really messed up the secondary structure.
Then that would affect your Raman.
I was going to say, just without better measurements, it's just sort of a guess at this point. Yeah.
If you're actually thinking that there's a non-specific binding, that's going to affect your Raman spectroscopy. So that's probably not going to give you enough of a like precise binding, or precise motif binding of the protein with your genome. That's
we should definitely talk about this later, perhaps. Yeah, I think that would be, sounds like you could possibly help us have some clarity on this. Yeah, that would be awesome. Thank you. Where are you located?
I am in the Center for Phage Technology at Texas A&M.
Oh, cool. Oh, awesome. Yeah.
Awesome. Thank you, Adriana. Okay, so next two questions. Quinan, you can ask yours, and then Vunkit Chin, you can ask yours. So Quinan, are you around?
Yeah. Well, let's see if I can. I just wanted to know, I'm really excited about the Phage, the plaque quant tool, as well as everybody. And I was just wondering what the base program or what the programming language is being used. Is it like being written from scratch, or is it like something that we can we can build from something like image J. So I was just wondering that.
Alina, are you on here? I don't see her name anywhere. Oh, shoot. I'm sorry, but I don't know. Yeah, I'll have to get back to you. I'm happy.
I think it's
oh, Brad.
Sorry, it's Brad. Hello. I think it's built in Python. And it's built from scratch.
But I think
Very cool.
she was going to create some sort of easier to use interface than just learning Python.
Yeah, so that's the main thing at the moment. It's quite difficult to use without a strong programming understanding. So yeah, that's what she's trying to do at the moment.
Awesome.
Yeah,
and she's compared and she's compared and contrasted it to some of the other tools, which I'm blanking on the name of it right now. It's like not Azure. Phage, that's the name. There's a Phage plugin that can do similar things, but it wasn't able to do some of the things she wanted to do, and so she decided to, write her own.
Well, hopefully there's a GUI or something.
Yeah, I think that's, like I said, I think that's what she's working on next, because obviously if you're just publishing some more raw code, then yeah, it's going to limit your number of people who can use it. But yeah, I'll shoot me an email if you're interested, and I'll let you know when it's, yeah.
Awesome.
When it's
Sure, thank you.
That's why it was in the soonish category, unfortunately. Cool. Yeah.
Vunkit, did you want to ask your question?
Hello, can you hear me?
Yeah.
I can.
Okay, so I was just wondering, have you done any actual studies yet on like those codon bias usages by the host bacteria? Because I read that X174 actually utilizes the host RNA polymerase. So I was wondering, have you done any studies on those?
Well, the codon, thanks for your question. So the codon usage pattern is very similar to the host.
Oh, okay.
Which is pretty normal, I think, because, they have to, well, I guess if they're going to use the DNA, the RNA polymerase from the host or the tRNAs from the host, like Phage's doesn't carry any of its own tRNAs, for instance, then it's going to have to sort of work within the constraints of the host. I tried in my changes to maintain sort of codon usage, abundance, CAI, I guess, codon adaptation index. I tried to maintain that within a fairly tight band. So if there was a choice between several codons, I would take one that was higher abundance, which is not always the best thing, especially if it's in the N-terminus. At the time I was doing this, which was quite a few years ago, it was a little bit less clear how important the certain codons might be in the N-terminus of a protein for translation. But I think that's much more clearly established now, that if there's certain rare codons at the N-terminus of a gene, you probably don't want to change those to a higher abundance one, especially. And so most of the changes, yeah, we're specifying a higher abundance tRNA if there was a question there. And the GC content was maintained across the whole genome, basically. So I didn't always just use Cs and Gs and things like that. Yeah, I think that answers most of your question.
Awesome, thank you. Okay, I want to ask a question from Fiona Whalen, who she would like it to be asked on her behalf. And I've also gotten someone else asking, please ask Fiona's question. So we're going to ask this one. Is the rational engineering of the ribosome binding strength being undertaken for the decompressed genome?
I did outline some plans for that, but that have never been done by anyone. I think finding the right person to do that's probably critical, but yeah, I mean, you could just imagine doing small library of a handful of ones for each of the positions, and just sort of seeing what happens to the plaque size in that sort of library, picking out larger plaques and sort of sequencing and things like that. So yeah, no, we haven't tackled that yet. Also, I was going to do, I think I had an undergraduate, I think it was last year, try to do some passaging of the decompressed phage as well, to select for higher growth phenotypes as well. So I like that approach. At least my one example of doing that was quite nice, in that it kind of showed us exactly what was wrong. And so, yeah, I think that would also be a nice experiment if I can convince anyone to do that, or maybe find the time to do it myself.
Awesome. Okay, two last questions.
Fiona.
Oh yeah, sorry about that. Two last questions. One of them, Jessica Neubauer, you want to ask yours, and then I'm going to ask another private one that came in, and then we're going to go to the breakout room.
Yeah, hi. I was just in general wondering if this kind of synthetic genomics is also done on archaeal phages or just bacteriophages, because obviously most bacteria are easier to grow than archaea because they can be weird sometimes.
That's a great question. I don't know anything about archaeal viruses or phage or whatever people call them. Yeah, I don't. I've ran across, a few papers here and there over the years and kind of been really interested. The only thing I know that they've been used for is like DNA origami and things like that. Or somebody I know was working on that for a while. I don't know if it ever went anywhere, but. Yeah, unfortunately, I don't know anything about them. Seems like some very green pastures there in terms of not much work being done. Yeah.
Yeah, I guess it's just because, with bacteriophages, you have E. coli that grows even if you don't want it to. And with archaea, you always have a bit of difficulty about thermophiles or halophiles or whatever files they have.
Awesome.
No, it's a cool
Thanks,
question.
great talk.
I don't know, maybe somebody can enlighten us, because I'd like to know. Yeah, I haven't ran across any examples of that.
Yeah.
And imagine some of the challenges would be very different, and especially in terms of, if it's a thermophile, like structure, GC content is probably extremely important under certain circumstances. Yeah.
Okay, thanks.
Awesome. Okay, last question from Mohammed Imam. He had to run early, but he wanted this one to be asked so he can watch it in the recording. About the phage PHAVES 174 that you've been studying, and he said he noticed it infects E. coli and Salmonella. And do you know the receptor, and is it the same receptor on both species?
It's a lipopolysaccharide. I was going to look this up before the talk, but I don't remember if the Salmonella lipopolysaccharide is identical, but there is some, there's got to be some similarity. Let me just find the slide where I talked about that. I'm not a real expert with the sugars. People at Macquarie are, though, so I am going to probably end up collaborating with them eventually. Just share my screen there. Yeah, so it's the terminal galactose and glucose for the R1. And so there is some variability because all of these different types can be infected by PHAVES, although the efficiency can be slightly different. But I didn't show the K12 form here, but it's not infectable. So if there's certain residues there, it's definitely cannot be infected. So the question is, yes, we know the receptor. It's lipopolysaccharide terminal residues on the outer core. And there's some that are known to be acceptable and some that aren't. Alina, the one who is my master's student who is working on that wonderful piece of software, her main project is actually looking at how can we modify the host range of PHAVES using mutations of the capsid proteins. And so that's some of the work she's doing. And so I think we'll learn more about that soon. Somewhat recently, I think there was a microviridae used in one of the SOREC labs. They do a lot of this cool CRISPR, anti-CRISPR work. They found a microviridae member that can infect K12 strains. And so we have that now from them, and so we want to understand a little bit more about that protein, and so how that difference in the protein might allow it to interact with a slightly different receptor. So stay tuned.
Yay, cool. Okay, so I'm going to take us over to the breakout rooms now, but first, thank you so much, Paul. That was extremely interesting, and I'm sure we would all just stay up and keep asking you, but we got to let these people go to bed. Get back to the lab if you can, if you're lucky enough to have an open lab. So I'm going to stop the recording, and the breakout rooms won't be recorded.